Hello, good afternoon or good morning, wherever you're watching us from. My name is Martin Winter. I'm the founder of Lab Automation Network here in Tübingen, Germany, and welcome to our webinar within the LAN Academy on temporal single-cell omics in cancer research. I'm very happy that you joined and to introduce you to our agenda. Today we will have first, of course, an introduction into the topic and also into Lab Automation Network, our organization, and what you will be hearing and seeing today. Then in about 10 to 15 minutes, we will have a presentation by Cytosearch, Tomasz Gereje, about LiveSeq, the temporal single-cell profiling with Fluid FM, a very special technology. You will see that later. And then in a second presentation, Anders Johansson from Qlucore, a company from Sweden, will present how to easily perform data analysis with the Qlucore Omics Explorer, which is makes it very easy to analyze very complex data sets like omics data without being a data scientist. And then we will have enough time for question and answers. You will find a button in your Zoom webinar window where you can edit your questions, and we will make sure that these questions will either be answered within this webinar or afterwards when we have more than we can discuss live. So please use this chat, not the chat button, but the Q&A button for such questions. All right, quick introduction into what we are doing at Lab Automation Network. We are an organization that combines the expertise of several expert companies in the field offering different technologies and solutions and expertise, and combine those solutions to very complex, if needed, lab automation and digitalization solutions, also special technologies like we see today. Our belief is that every challenge in the lab should be met with the most effective technology and expertise available. And our mission is to deeply understand, as we are a team of scientists here in Tübingen in the LAN team, to understand the unique requirements of our customers and give the users in the lab unrestricted access to the most advanced and proven lab automation solutions. That's what we are doing. And we're doing this along the customer journey. So we're offering solutions, trying to match your requirements with the best technologies and solutions. We're doing this, by the way, free of charge, so no consulting fees, because we are paid by our technology and solution providers. And you finally, if you choose to implement something, you make this deal directly with those suppliers. What we offer in addition to that is engineering. So we do things like user requirement specifications, feasibility testing, simulations, before you finally decide to implement a lab automation or a digitalization solution in your lab. We help you with implementing this with project management. And also, and that's what we're in particular doing today, we are offering courses, webinars, hands-on workshops on different topics and technologies. And today's webinar on single-cell omics is one example of exactly that. So we cover the entire customer journey with education, planning and de-risking projects, building solutions, and finally implementing them. This is a quick overview of some of our partners. You will also find, of course, Qlucore and Cytosearch here on this list. There are many more, also systems integrators and robotics companies, in order to let you know how these solutions can look like. Just a couple of images. And this in particular is a visualization of the single cell extraction technology that we're going to see today. And of course, also software technologies. So to start with our topic of today, with our wonderful speakers, what if you could track single cell transcriptomic changes over time without killing a cell? This is a question that we want you to answer, maybe. Yeah. Could you imagine doing this without destroying a cell? And then if you have acquired the data, the transcriptomic data, for example, then what if you could analyze these complex omics data effortlessly without being a data scientist and without coding expertise? So, for example, if you're not familiar with R or similar instruments or similar technologies and software. So our speakers today who will present these technologies is first Tomasz Gerecei. He's a biophysicist and has a robust background in single cell manipulation technology. He obtained his PhD in biophysics from Eötvös Loránd University in Hungary, and is working with CytoSearch from Switzerland in Zurich. And he is an expert for the fluid FM technology, which he will present you in his talk. Then second speaker later on will be Anders Johansson. Anders is an experienced professional in bioinformatics. He is currently sales director at Qlucore, a company from Lund in Sweden. And he there leads the sales for bioinformatics software solutions and is helping researchers from different domains to analyze complex data. Before that, he worked at Applied Materials, Aquanova, EU Metrics, and other companies in similar areas. So let me switch to our survey that we would like to start right now. And because we would like to more focus on what's really of interest for you today. So you can, in a minute, answer those questions in our survey. First question would be, what is your biggest challenge in single cell transcriptomics research? And the second question, before today's webinar, how familiar were you with these technologies, being LiveSeq from CytoSearch and Omics Explorer from Qlucore? And we will now start the survey, so you can tick the boxes that apply to you. So first question, what is your biggest challenge in single cell transcriptomics right now? You can also choose several boxes. I let you read the individual options. And then the second question is, before today's webinar, how familiar were you with these technologies? Also here, you can select different options. The third option, of course, excludes the first two. If you have heard of LiveSeq, of the Omics Explorer, or never heard of either before. And if we have collected all the answers, we can share the results and see what you've answered. So here we see the results. So most of you answered the first question with difficulty integrating different omics datasets, and also destructive sampling methods that limit longitudinal studies. These were the most important answers to the first question. Thank you for that. And the second question, never heard of either before, and that's exactly why we are here, to show you what these technologies can do for you. All right. So this should be good for our first talk to Tomasz, focusing on LiveSeq and the temporal single cell profiling with Fluid FM. And by this, I hand over to Tomasz. Please share your screen and start your talk. Welcome to the webinar.
Hi, Martin. Thank you very much for that nice introduction. So yes, I will share my screen, and you should be seeing that now, hopefully.
Yes, perfect.
All right. So indeed, what I would like to talk to everyone today about is something that is called temporal transcriptomics, and it's very new because, as it also turns out from the quiz, transcriptomics is limited by being an endpoint measurement and really missing out on the temporal aspect, which can be very important in answering certain biological questions. So I would like to actually spend a bit more time on why to do this and what this brings to biology. And then I will, of course, also talk a bit about how it is done using fluidic force microscopy. So to understand the why aspect of what these temporal studies can bring to the table, it's really nice illustration to bring up this experiment from the proof of concept paper of LiveSeq, whose reference is down here. I really encourage everyone to read this paper. It's amazing, and yeah, it just really encompasses everything that this is about. And to really understand this experiment, let's start with the system that the authors were using here. So this was a pretty simple cell line with a reporter inside of it. So this is a macrophage cell line called row. And in this macrophage cell line, there is an M-cherry expression that is driven by the TNF-alpha promoter. And in this certain cell type, this promoter is related to the inflammatory response, because macrophages are indeed immune cells, of course, and they go to the side of inflammation when they receive a certain inflammatory signal. And this, of course, changes their transcriptome. And in this kind of cell types, this kind of cell line, this is read out in a one-dimensional way through fluorescent imaging. And that's why this is really useful for us in this case. And what we see then through simple fluorescent microscopy is that if you look at these cells, then before any kind of inflammatory signal would be applied in the sample, they are dark, so not expressing fluorescence, so we can see here. And once we would add the LPS to the sample, which is it's a bacterial molecule that is commonly used to induce inflammatory conditions, then we can see that the cells start to react and they light up in red. So this is the readout of their phenotype. And we know that the LPS induces a transcriptomic change as well. And why this is a really good system and why I went into so much detail about it is that it really encompasses a general experience in biology, which is a large heterogeneity on the single cell level. So if you look at these curves, we can already see that there are some cells that are extremely strongly expressing. There are some that are a bit less, and that there are some that are in the average. And this is a general experience. Of course, everybody who would deal with single cells will see that even though these cells are genetically identical and they're in the same environment, they will have a very different reaction to external cues. And when we want to understand such facts, then we can ask, okay, well, this has to be some kind of gene expression difference. So what is driving this gene expression difference? And to answer that question, a really easy experiment, if you don't really have any technological barriers, would be the following. We can take these cells, the individual cells, and then measure their full transcriptome somehow. And of course, that is normally RNA-seq. There are different RNA-seq techniques that can determine the transcriptome, but let's just imagine one way or another, we determine the transcriptome, and then let's say we could do this LPS treatment on the same cells, to induce the inflammatory conditions. Then we read out the M-cherry expression. So we are measuring the fluorescence. And if we have really all of these informations together, so we have the phenotype characterized to fluorescence, and we also have the transcriptome because we measured that at the beginning, then we can simply correlate these two things. And then we can see which genes change together with the phenotypical response. And this is indeed what the authors could do. So here you can see a list of most variable genes in terms of these genes expression changes together with the phenotype. And they could see some genes whose expression was really strongly anti-correlating with this response. That was not really revealed before. So why is this a big deal? And it sounds really simple, of course, but if you look a bit deeper into this experimental flow here, it seems impossible, because once we measure the full transcriptome of a cell, which again is RNA-seq normally, that's a destructive technique. So then the cell is dead. It doesn't exist anymore. Normally we have to lyse the cells to access the RNA, and then they are not there. Therefore, it's impossible to do the LPS treatment, M-cherry expression measurement, because we destroy the sample. And so this kind of experiment, and this is why this was such a big deal, is still is only possible if we manage to sequence cells without killing them. And this is really the power of LiveSeq or live cell sequencing, as also the name suggests. All right, so how is it done? Is very simple. So before the full transcriptome measurement, we implement another step, which is what we call the cytoplasmic biopsy collection. And this really means that we go into the cytoplasm of a cell with a little needle, which is the fluidic force microscopy that I will explain in a minute. We go into the cytoplasm and then literally take a biopsy from the cytoplasm of the cell. And similar to a biopsy in hospital, the patient should survive this, which in this case is the cell itself. And it does in most of the cases. So that means that instead of destroying the entire cell, we take a sample, and then actually is this sample that we are going to sequence. And then the cell is still there in the sample, and it's possible to do different treatments and to measure the phenotype, which of course can be different things depending on the different biological system. And so in this concrete example, just to reiterate on the kind of data that could be measured in this paper, is that here you would have the cells, which are each data point is one cell here. And what you can see is that there are two different kinds of information for each cell. First, there's the gene expression before the treatment. So this is like is the state of the cell when the treatment hit. And then we also have for the same individual single cells, the response to the treatment. And then you can discover correlations. So this really shows the possibility to have a certain cell state measured, and then, so temporally after that, the reaction of that cell to an external stimulus. And again, this is of course only possible because to measure this piece of information, we didn't have to destroy the cell. And then if you don't have to kill the cell for sequencing, then of course that kind of also implies that if you find that cell again, which of course that's very possible to do, then you can also sequence it multiple times. And this is also something that was achieved in this paper. In the same system, we can see it here. So this cluster represents these macrophages before any kind of treatment was applied. So this is the ground state. And the dots are this normal RNA-seq. So those are endpoint measurements. And then this is the only point we have, right? We don't know how that individual cell would evolve. Then we can do the same for treated sample. So that would bring the cluster here. And of course, again, these are different cells. However, the arrows here, they really show the LiveSeq data. So that means that we have two different transcriptomes for the same single cell. In this case, 4 hours has passed between these biopsies and the transcriptome. So this really shows that it's possible to not just look at one transcriptome cell, but how it changes dynamically. Conceptually, it is also possible to take more biopsies, change the timelines more for longer terms, and look at longer time evolution of these cells. So, of course, another case where it is very interesting how cells change their transcriptomes. It's not just thanks to, let's say, phenotypical reactions, but also complete cell transformations, which is called differentiation, for example. And this was another experiment in this paper that I will not go through in much detail, but again, I really encourage everyone to look at this separately. Is done with these adipocyte progenitor cells that have like a well-defined differentiation pathway. And the authors could also take biopsies before and after differentiation from the same cell. And here it's important to note that there was 2 days between biopsies, so you can see how the time scale can be adopted to what that biological process is relevant for time scale wise. And so, yeah, indeed, here we also have these time-resolved transcriptomes, which, again, this is kind of a proof of concept. So this system is a bit more straightforward. However, of course, there are other progenitor cell systems where there are many different downstream pathways of differentiation. And this is where we expect this technology to be very, very useful to determine a certain cell would go down on a pathway for what reason. What are these transcriptomic signatures that would predetermine the cell to go a certain way in its transcriptomic journey or cell fate, really. And so this is going to be a method of choice for developing these theories. So how it's done in a bit more detail on the technical end. So of course, then the data analysis is what will be the second part of the webinar, but also I just wanted to mention on a physical level what happens, because it's also really interesting how these biopsies get taken. So this is based on fluidic force microscopy, as I mentioned before, so FluidFM, which is the patented technology of CytoSurge, which is our company. And just to give an overview, of course, there's like a lot of details in there, but the point is that the cells are going to be sitting in a 2D sample. So it's important to know that this works on like cell lines or potentially also primary cells, but they need to be in a 2D sample, which can be any kind of petri dish or well. And they are targeted by this FluidFM cantilever, which in this specific one, we call this a nano syringe, because it's like syringe, but nano. And the way it works is that we basically can point at a certain cell from which we need to take the biopsy from. Then the cantilever will approach this cell, and thanks to a force control mechanism, it can pierce the membrane in a very gentle way, which means that we are always telling the cantilever what kind of forces to apply when it pierces the cell membrane. So is it 200 nanonewton, 300 nanonewton, and this allows it to be very gentle. And then once we are in the cell with this syringe, then we can apply negative pressure in a channel which is inside of the nanosyringe to literally suck out the cytoplasm. So it just applies like a suction force thanks to negative pressure. And then, as you can see here, the cytoplasm goes into this channel, which is in the middle, but which goes through the entire length of the cantilever itself. And here behind the cytoplasm, there is oil. So it kind of works like a piston that it brings out the liquid cytoplasm, but there is no mixing into what is already in the channel. And this volume is 1 to 2 picoliters. So it's extremely small. It's not really compatible with anything. So you cannot do much with 1 picoliters. And that's why we have to deposit it into a droplet. And this is really the output of the experiment. This is a 1 microliter droplet that contains the RNA together with the buffer. And then this is something that can be taken for a library prep and then sequencing. And then this library prep has to be an ultra low input library prep, for which there are different techniques, but we have some good results with the Qlucore chemistry by Nexogen, for example. But yeah, we can also, of course, give some advice there. And just as a last, I will skip this one, and just as a last conclusion, I want to also show you how this is adapted in the laboratory. So if somebody wants to do this LiveSeq technology, then the first thing they need is the FluidFM Omnium, which is a laboratory device that looks like this. So it's similar size to a microscope. It has an integrated fluorescent microscope. sample stage and the fluid FM head, like the measurement part, comes from the top. And this is how the nanosyringe itself looks like. So you can see how sharp, what the sharp tip is, and then it goes into the cell and then applies a negative pressure to take the cytoplasm, which will then flow through this little hole. Of course, we can also reverse the actual workflow and inject stuff into the cell, but that would be a separate webinar, which we should have maybe one day. So right, this is pretty much how to get started. There is, of course, a lot of details there that we are also happy to talk about later on. All right, so with that, Martin, I can give you back the word.
Thank you very much, Tomasz. Very good talk. And we also have the first question here.
All
Maybe
right.
we have a couple of minutes for technical questions. So this first question is, great talk. How can we be sure that the microbiopsy is representative of the whole cell transcriptome?
Well, yeah, that's a very key question, of course. So what you have to keep in mind is that this 1 to 2 picoliters, it's small in absolute terms, but in relative terms, this is 30, even up to 70% of the entire cytoplasm. So it's a big sample. And that's one indication that it is supposed to kind of represent, at least physically, the cell. And the better indication is that this was more or less examined in the LiveSeq paper. So this original proof of concept, where they integrated these LiveSeq data sets into like normal RNA-seq data sets. And they fit in perfectly. So it seems that the data that you get is very much from what you would get from normal single-cell RNA-seq. So as much as that is representative, this should also be. But it's true that there can be like very polarized cells. So for example, neurons is something that we will start working on soon. And there this might not be the case anymore, because there, of course, there is like a large polarization of the cell, which is well known.
Okay. Thank you very much. And to all the participants, please don't hesitate. Put your questions into the Q&A section. There's a follow-up question from the same participant. If we remove so much cytoplasm, the cell biology is not affected?
Good question. It is affected to some degree. So what has been demonstrated very nicely is that the cell cycle is affected in the first round. So basically that cell cycle in which the biopsy was taken will take longer, because the cell needs time to replenish its cytoplasm. And this cell cycle disturbance, this also comes with a very limited transcriptomic signature, which was very nicely described in the paper. So it does have a limited effect, but not enough to really disturb the entire transcriptomic journey that we are looking at.
Okay, thank you very much, Tomasz. And then I'd like to hand over to Anders Johansson. Anders, stage is yours.
Thank you, Martin. Hi, everybody. So let's see, get the screen shared. So I wanted us to start here with a couple of words on Qlucore, who we are and where we come from. And then I will also run a demonstration on the same data or experiment that Tomasz presented here. So we are a company in south of Sweden, Qlucore, focusing on the analysis of biological data, both in research and diagnostics. So the company have two different legs that we're servicing, both research and scientists that wants to have a visual approach to data and analytics. And we've been around doing this since 2007 and has a large presence now in academia and industry in life science. We also, since couple of years back, have a second leg where we support clinicians with machine learning classifiers to help the diagnosis of so far oncology patients. So we have brought our leukemia model now to IVDR approval, and now more models coming as we speak. But today's webinar and demonstration will be focusing on the product Qlucore Omics Explorer for exploratory data analysis. So the product Qlucore Omics Explorer there is a tool that's been developed to enable anyone, both advanced, let's say, mathematical, statistical experts and people without that background of mathematics and coding, to have a visual relationship to their data and being able to explore data. So we want to cater to the biological subject matter experts that wants to analyze their own data and thereby increase innovation and further knowledge. So in order to prove this, I wanted to run a demonstration here in our software using the same paper that Tamas referenced here before, and especially then in addition to that, the data from this article is downloadable from the Gene Expression Omnibus, the GEO database. So this data was downloaded in matrix format and imported to Qlucore Omics Explorer. So what I wanted us to just have a look at is to visualize, perform a so-called variance filtration to reduce noise in the data. Visualize this, the transcriptomic profile of different cell types that was used in this one. That's a simple exercise of visualization. Then we will also have a look at the LPS treatment that Tamas explained there, where macrophages were treated with lipopolysaccharide and then matured and then we will look at find the 70 or so most discriminating genes for this transformation and visualize this. So here I wanted just to make a disclaimer. I have never been involved in this article and came across this data and found that it was a good example here to show this demonstration in this context. So let's move over to the Qlucore Omics Explorer. So here I have the data is always visualized in a PCA plot, and the data set, the connection to the data set is by choosing plots once you have several of them up. So what we will do now is to look at this data and emphasize, increase the understanding of the data by coloring. So this is all the data, and what I wanted us to do first is to switch and look at the different cell types that was used in these several experiments, I should say. So there are 4 different cell types, and if we want to visualize this, we can color according to 2 cell types. But in this case, it doesn't look so, they're not so distinguished, but we have in this data set, as you can see down to the left, 1,000 samples and more than 30,000 variables. So we can expect that there will be a lot of noise. One of the technologies in this situation is to perform so-called variance filtration, where we will take away the variables that don't fulfill the requirement we put on the standard deviation. So I will perform this by sliding this further and further to the right, and let's aim for, let's go down and see how the data updates as we approach maybe 1,000 variables or so. A little bit too far. So after performing this, now having 420 variables that are then adding, fulfill this requirement on the standard deviation, we see that we have good separation from the different, let's have a look at the different cell types here. So now they separate based on their transcriptome. That's not such a big surprise that they would. What I also wanted to show here is after performing this, the principal PCA plot is how Qlucore always present data. We can, of course, also add other plots and types. So as one example, I wanted us to have a quick look on how it will look when we choose the Tisney plot, for example, with the algorithm inside there to both perform unsupervised classification and visualize the results. And also this plot can, exactly like the previous one there, be colored according to the different annotations here. So here we can see the separation between the different cell types, and I can also put them nicely next to each other. So this was one example how you can then easily import and visualize raw data. What I wanted us to do next is to look a little bit more into detail on the experiment that Tamas showed us before, where they had taken this so-called raw 264.7, which are the microphages that were seeing this LPS treatment. So apologies there, we will start with a new plot and start all over. And as said before, we will start with the PCA plot. And in this experiment here, we are only going to look for the microphages. So when I unclick these ones, I take away these samples that are connected to this annotation, and they disappear both from the plot and any calculations in the background. So now we will switch to the treatment here and look at how the data changes upon LPS treatment. So the other two experiments there with the adipose cell, we take them away, and we only focus now on the before treatment and after treatment, and we color accordingly. In this case, then it's not so good for the eye to see the different colors. So let's make this choose colors that are very distinct. Yeah, blue, and then maybe red. So now the plot, and instead of variance filtration, this time I wanted us to look for the most discriminating genes between these two before and after treatment. So under statistics here, we will, for this purpose, yeah, there are several statistical tests to choose from. We will now choose a two-group t-test here and perform this test, set this up between the LPS treated versus the not treated. And as I perform this now, we are taking away variables, but not according to standard deviation, but the corrected p-value, which we call the so-called Q value. So I will keep sliding this slider over until I have aiming for the 70 or so most discriminating genes. And as we come closer and closer, we see that the before and after treatment will separate along the first so-called principal component axis. And this is a way to visualize data, the PCA plot, going from a multi-dimension, all the 30,000 going down to only 3 dimensions. So samples that are close to each other have a similar gene expression, and ones that are far apart. And looking at this plot, we can see what Thomas also said, that it's known that some of them mature and some don't. But we have a clear separation between two groups here. And that is the prerequisite for us then to do this type of discriminant analysis. So let's just quickly switch from this sample annotation and have a look at the little bit too tough here. I wanted to have. Let's go for 80. So here we can have a look on our variables. These are then our 80 most discriminative and we can add now the calculated statistics, and we can sort according to the corrected p-value. And we now see that here we have the most discriminating one, and then we can scroll down and look at this. Most commonly, this type of data is not visualized in a PCA plot, but rather in a heat map. So I wanted us to quickly switch to have a look at these variables now in a heat map. Let's auto-size this to the available window. Maybe we could zoom in a little bit here, and we can see that we have the sample names here on the x-axis, and then the variable names on the y-axis. Auto-sizing this, and then we can order them hierarchically, both the variables and the samples. And then for further visualization, we can color according to the annotation here, which was treatment. So we can see now that we have both in the what did we have? We had 80 variables here being visualized, and most of them are upregulated as a result of the LPS treatment. Yeah, red was LPS treatment, so most of them, but also a few that has been downregulated. So this was a short demo on how to explore data, how to visualize, find discriminating variables after a statistical test. Then one possible step after this could then be to continue using the gene set enrichment analysis in the Qlucore software to find what biological pathways have been affected by this LPS treatment, and also then in each pathway find the core enriched genes. And so this is one possible follow-up on this. If you got interested in learning more, I wanted to just highlight that we have weekly webinars and training courses in the usage of Qlucore Omics Explorer, and you can also download your free trial and contact us for support or help, or join any of our online training courses. So with that, I open for questions then.
Thank you very much, Anders. Very nice presentation and perfectly in time. Thanks for that. Tomasz, please rejoin for the Q&A session. Anders, what you just showed us and demonstrated, was that live or pre-recorded? Did your computer calculate all these results live?
No. I actually performed the analysis as we went along. So yeah. So when I run those, for example, the statistical test there on the slider, the calculation engine in the software updates the model, background model, and the plot close to real time, even if we had 1,000 samples and what was it, 30,000 variables.
Mm-hmm. Okay. And in addition to omics data, is it also possible to use mass spec data and other types of complex data sets?
Yeah, any multivariate data can be imported and visualized in Qlucore Omics Explorer. Other omics data like proteomics and metabolomics coming from mass spec are many customers use. But with that said, I want to emphasize us as a company, and the software is developed towards biological data. So.
Okay. And we got one question associated to this. Is it possible to merge two different data sets and get insights? For example, single cell transcriptomic and proteomics data collected on different data under similar conditions.
Yeah, correct. And then look for correlation between the different data sets is one application of this. Yes.
Okay. So please, questions are open. Tomasz, a question for you. what kind of cell or what cell lines, what type of cell lines can be biopsied with your technology?
Yeah, so there have been a few cell types tested that are mostly like cancer cells. So for sure, those are a bit more robust and we can work with them. But in general, any adherent cell type should be possible to work with. So we can for sure pierce them. And then the viability on all the cell types that have been tested so far is at least 80%. So it means that the cells are really resilient most of the times.
Yeah, and then we have a question in the chat window. Let me see if I can see it. Just a second. Who are the target users for this application? I think this question goes to Tomasz.
Well, for ours, yeah, it's probably basic researchers. So we think that this is going to be a really game-changing technology for basic research. So, for example, in terms of cancer, which is what we were talking about just now, it's for example something to discover ways of drug resistance. So one phenotype that could be read out is just a simple, did the cell die or not? And then the treatment, instead of inflammation, could be like a chemotherapy or some kind of anti-cancer drug. And then that would make LiveSeq would make it possible to actually look at which transcriptomic signatures predetermine drug resistance, because then we can check which cells died and which didn't, and then look back, okay, what was the transcriptome of this or that cell before the treatment? So, for example, that's why we are really excited about this application, to promote it to cancer researchers, because these kind of questions can be answered.
Yeah. Then another question for you, Tomasz. Have you tried this technology for multinucleated cells like muscle cells and osteoblasts?
Yeah, well, not really, but I mean, that should work. So the thing is that the bigger the cell, the better, actually. So we are really sure that if we're looking at like an enormous multinucleated cell, that would be even like an ideal target for this technology.
Yeah. Okay. If you have further questions, please put them in the chat. In the meantime, we would like to ask you a question. Let me quickly share the question here. So the question we would like to ask you is, after this webinar today, how do you see the potential of LiveSeq and intuitive omics analysis in your research? So if you like, you will now see a question window opening. Hopefully, here it comes. So very promising to see direct applications. Interesting, but need more details. Or if you're more neutral, relevant but unsure how to apply it, or not very applicable to your research, or still have questions, like to follow up, you can choose and give us your opinion. And of course, independently from your answer here, we are always open to take your questions also after the webinar. And we all, Anders and Tomasz, are willing and able to contact you in detail and discuss in detail all of your open questions. And if we have collected your answers, maybe we can show what's coming out. Okay, very promising. 3 votes. Interesting, but need more details, 75%. Okay, that looks promising. So please ask your questions. If you have any question about details that you would like us to answer right now, put them in the Q&A box. Otherwise, send it to us after the webinar and we will follow up with our answers. Yeah. So, as I know, Anders, you are traveling also and having seminars in different places throughout
Absolutely.
Europe. What is your next stop for an Omics Explorer seminar?
Come next week, we will give a Qlucore Omics introduction training course at the University of Basel. So that will be the next out in Europe event coming. And then we keep these, we publish these events there on our LinkedIn page. So feel free to follow us there and get an update on our different local events that we run.
Yeah, and in Switzerland, I think, Tomasz, it's also possible to see the technology live.
Yeah, absolutely. So we are always happy to receive visitors in sunny Zurich.
Yeah, it's in Gladbach, close to the airport, right?
Exactly. And not really sunny, but yes, yeah. Happy to do demos, and not just that, but if someone has like different cell types for which the technology has not been tested yet, then we are also happy to collaborate and test like different cell types, whether they are resilient against a biopsy. Can we generate libraries from that? So we are really open to collaborating on such projects to generate proof of concept data.
Yeah, perfect. All right. With this, I'd like to close the webinar today. Thank you to all the participants. Thank you, Anders and Tomasz, for your presentations. And also thanks to my team and both of the Stephanies who prepared and led this webinar technically. So thanks, everybody. Have a nice afternoon and a nice rest of the day. Bye-bye.
Yeah, thanks. Bye.